A very good evening to all of you, and it gives me immense pleasure to again come back with the Rare Info Series. And today we have Professor Arijit Mukhopadhyay, who also happens to be my senior during, like my super senior during my PhD years. And it's a great pleasure to welcome him to this Rare Info Series. Arijit has been passionately working on glaucoma. From bench to bedside, as I must say. And he is a professor at University of Salford in precision medicine. And without wasting much time, I will pass it to him so that he can give you his lecture, which he is so passionate about. Thank you, Arijit. Welcome you to the Rare Info series.
Thank you, Dipanjana. Thank you, Prasannaji and the ORDI team, Chetna and everyone for making it happen, including the initial technical glitch. I was not able to share my slides, so thank you for everything. As Dipanjana said, I am working on this field, genetics of glaucoma and how genetics can change lives for better, has been one of my longest standing passions in my scientific career. I currently work in the University of Salford, which is in Manchester in the UK. But the work I'm going to share with you today is largely happened in India. It started in India and now still continuing with partners in India. ORDI is a partner in this journey, as well as Dipanjana. And many others. I'll show some of them in the talk. If I can move to the next slide, please. So glaucoma is a disease. Many of you might be familiar with the name and you know what happens. So if you just click once more, you will see the field of your vision diminishes. Just click once more on the slide. So it reduces your field of vision. So peripheral vision decreases like that, and it becomes a tunnel vision. And the most dangerous thing in glaucoma is most forms of glaucoma is that it is painless. There's no symptom. You don't really realize that you are losing vision until you lose about 70% of the sight. So that's really what we are dealing with here. Next slide, please. Glaucoma is a complex disease, like any other complex disease you can think about. Think of asthma, think of diabetes, think of cardiovascular disease. It is complex. And there are many subtypes, again, like any complex disease. The subtypes that are primary, so you can see on the slide, there's a primary and the secondary. The primary subtypes are largely genetically linked. Some of them are directly linked, so it's a monogenic condition. Some of them are multifactorial condition. Secondary glaucoma are usually not genetic, or it is genetic but for another comorbid condition. For example, a non-genetic example of secondary glaucoma would be if you get a trauma in your eye. Let's say you get hit by something in your eye by an accident, that can turn into glaucoma. So that would be a secondary glaucoma, which would not be a genetic form of glaucoma. So today we'll talk about primary forms of glaucoma. And within primary, there are three subtypes again underneath primary. There is primary open angle glaucoma, POAG, which is on the left side box. Then primary congenital glaucoma, which happens at birth, which is called PCG. And then there is primary angle closure glaucoma. That is much more complex genetically. So out of these three, if you click once more on the slide, so glaucoma is fairly common. As you can see, it's more than 100 million people globally are affected with this condition. And this is likely to be an underestimate. It's probably much more than that. And if you click once more, you will see that POAG is about half of them. So primary open angle glaucoma is the largest subtype of the parts of glaucoma that are largely genetically caused. And it is less common than cataract, but it is the second largest cause of blindness in the world. If you think of blindness that is irreversible, so cataract is largest cause, but it is reversible. In most cases, you can do a cataract surgery and you can get your vision back. But if you look at irreversible blindness, then glaucoma is the largest cause of irreversible blindness. So I'm going to talk about that form, POAG. Thank you. And so you might wonder that why, if this is so common, why I'm talking about this in a rare disease rare info series. The reason is POAG and other forms of glaucoma as well, but I'll be talking about POAG from this point on, has two completely different genetic mechanisms of a disease manifestation. The one side, which is showed in green, are the rare forms. That's only about 10 to 15% of the total disease burden. And the more common form, 85 to 90%, is a multifactorial, more common kind of disease. That's far more complex, right? And the genes that are named, there are 6 genes you can see named. There are 20 different chromosomal regions mapped for the rarer forms of glaucoma. Which are more genetic, like any other rare disease you might be familiar with. It is not as rare as other rare diseases. So largely rare diseases are typically 1 in 2,000 live birth or something like that. These genetic forms of glaucoma are Mendelian forms of glaucoma are about 1 in 800 or so, right? But the reason I'm talking about this in the Rare Info series is the same principles apply, and for the same reasons why rare disease patients can be helped using genetics and precision health approaches, the same concept, same principles applies to this rare component of glaucoma as well. Hence, this session is dedicated to that. And so that's the focus. And as it says on the slide, there are no genetic overlap between the two forms. So what I mean by that is the genes that are found to have mutation for the monogenic forms of glaucoma are not found when you do genome-wide association study type of work for glaucoma. So there's no overlap between multifactorial type and monogenic type. There's one of the genes called myocilin, MYOC, which is in a red box on the slide, because the main data I'm going to show and the project I'm going to talk about is based on that particular gene. Next slide, please. So we have been working on this for a long time. It's about 2 decades I'm working on glaucoma genetics. And you can see a summary at the bottom of the slide. So I have 22 publications so far on glaucoma genetics. We found 3 new genes along this journey. I had a TED talk on this story that I'm going to share today. It has inspired one fiction novel, which I did not write. A fiction author writer wrote it. I'm not going to talk about that. And I also produced 2 films based on this work. I'll talk about it a little bit more later. And as you can see on the slide, I just showed you just 6 papers as a snapshot out of these 22 papers, just to show that we have addressed both types of glaucoma. We have looked at the rare form, which is on the blue, labeled as blue, and which is also looked at the common form. So we have done exome sequencing, we have done single gene sequencing, we have also done genome-wide association studies, we have looked into mitochondrial genome variations, we have looked into deletions like copy number variations. So we have, when I'm saying that this and this form and this form, the complex form and the monogenic form does not overlap, I'm talking from a large body of work that we already published from that experience. Next slide, please. So this is just an example of a paper that we recently published. Here we have looked at only on familial forms of glaucoma. And what you can see on the slide is just a snapshot of pedigrees, that what kind of families we worked on, just to make sure that we are looking at rarer forms of the disease. We are looking at rarer mutations with a higher penetrance. If you're not familiar with the term, higher penetrance means a mutation that has a large contribution to the disease manifestation. So on the left of the slide, the pedigrees are from families affected with open angle glaucoma, and on the right, the families are affected with angle closure glaucoma. And what we have shown in this paper is that we identified two new genes doing whole exome sequencing of these families. And we also showed that although clinically the open angle glaucoma and closed angle glaucoma are different, but when you look at the molecular signature, when you look at the whole exome kind of a profile, there are considerable overlap in the pathway of the genes where we found the mutations, right? So that's an example how we approach the rare form of the disease. Next slide, please. Another example where we looked at a single gene duplication and where we showed that within POAG, there are patients who have high eye pressure. We call them high tension glaucoma or HTG on the slide. And there are patients who never develop high pressure, but they still have severe damage due to glaucoma. So they are called normal tension glaucoma or NTG. So this particular duplication in TBK1 gene, for example, again, a rare form of the disease, but it is specific for normal tension glaucoma. It's about 2% of all glaucoma patients have this particular duplication. What you see on the slide, which is circled, the two blue dots are circled on the top on the NTG box. So every other dot is below, and that's the copy number of the TBK1 gene at the genome level, at the DNA level, but only for about 2% of the NTG patients have this duplication. So again, this can be a biomarker. So you can simply screen for this duplication. There is a Tachman assay specific for the duplication, and you can diagnose somebody even before they show symptom as a normal tension glaucoma, a pre-symptomatic individual. And this is not only our work, this has been shown globally that it is consistently about 2% of glaucoma patients, and is always this duplication are directly linked to normal tension glaucoma. So this is again another example how we can use precision health or precision genomics. It's a precise information to in order to allow us for a precision diagnosis and then a precise management. Next slide, please. So coming to myocilin gene, as I said that I'll be focusing on this particular gene most of the lecture. So out of these rarer forms of glaucoma which are genetically caused, which is about 10% of all glaucoma, but 90% of that 10% is caused by mutations in one gene, which is myocilin. So that is obviously the most important candidate that we always screen. It's a tiny gene from a genomic context. It's only 17 kb at the DNA level, is only about 1500 base pairs at the cDNA level or the RNA. And what we showed long time ago, that there are only 20 nucleotides, which is shown as two vertical black bars within exon 3 of this gene. There are 20 nucleotides that accounts for 30% of all mutations in the gene, right? So they are like mutational hotspots. And so we have published this, and I'm going to talk to you about a particular family in West Bengal who happen to have a mutation in one of these 20 nucleotides, and how we devastated their life and how we are trying to reconstruct their life for better. Next slide, please. So this is a case study. So the boring string of letters you are seeing on the slide is a sequence from myocilin gene exon 3. And the highlighted part on the slide, CCG, is a codon that codes for an amino acid called proline. If you click once more, so we found a family where one of the letters is changed, a C is changed to a T, and the CTG now codes for a leucine. So what should have been a proline, it's now will code for a leucine. And this happened in this family. I'm going to show you the family in a minute. This happened in this family in heterozygous form, meaning what? Meaning you will inherit 2 copies of this from each of your parents. This is in chromosome number 1. So you will inherit 2 copies from each of your parent. So when I say heterozygous, it means for those who have this mutant allele, the T letter instead of the C letter, they will have the T in one of the copies. The other copy is C as expected, as normal. Next slide, please. So we found this mutation, and this is the picture of the eye of that particular patient, proband, the index patient from this family. And again, as you can see, we published this a long time ago. So you can't, if you are not an expert or an ophthalmologist, you will not see what is wrong in this picture. So if you see the central part that is bright in the eye, so that's the place where the light falls, and in the central part where the nerves go out in the back of your brain, the optic nerves, and there's a thinner layer within this bright area, which is yellow layer, that should be thicker. It's thinner in this particular patient. It is thinner. The reason is that most of the nerves and neuronal cells are gone, are lost, they're dead, because this is a severe glaucoma phenotype in this particular eye. So they don't have many neurons left that can carry the signal back to the brain. So this was the patient we saw more than 20 years ago. Next slide, please. And we found the mutation. I already showed you the mutation. I told you it's a C2T mutation. So that's the more of a molecular context. So on the top panel, what you are seeing is the schematic. So it's an exon 3. And I already said that it is CCG codon changed to a CTG codon. And that changes a proline to a leucine at the 370th amino acid of the protein. And in the middle panel, you see a chromatogram. If you are familiar with this kind of visual, then you see for the position the pointed by an arrow, you see 2 colors instead of 1 color everywhere else. So that shows that the 2 alleles, one from the mom and one from dad, are having, one is having a C, one is having a T. And we also developed a simpler assay where you can use an enzyme to cut the DNA depending on whether it has the T allele or the C allele. So typically we are able to cut it in 2 fragments, which you see in the bottom panel, if it has 1 C allele and 1 T allele, and if it does not cut into 2 fragments if it does not have the T allele. So this also we published more than 20 years ago. So it was a hotspot, very well-known mutation. It happened to be always associated with aggressive forms of glaucoma. So we knew all that. So this potentially put us in a position 20 years ago where we could tell anybody, any child at birth, before they have any problem in their eye, that this individual is going to be blind and this individual is not going to be blind. But that it didn't turn out that way. Please go forward. So this was the family, right? I guess the people in the audience are familiar with how to read a pedigree. If you are not, I'm just pointing out the key features. So a square is a male, a circle is a female. If it's filled in with a black color, that means that person has glaucoma in this family. And if you see a line, a slant, like a forward slash going through the symbol, that means at the point of collection, at the point of this pedigree information, that person was no longer alive. And the generations from 1 to 4, so the people drawn at the bottom of the pedigree are the younger generation, at the top they are the older generation, right? And the individual with a bold black arrow is the individual for whose eye picture I showed a couple of slides back. So that's the proband, that's the index patient, right? So this was, for those who are familiar with reading this kind of pedigrees, this is an autosomal dominant form of inheritance, which is a classical textbook form of autosomal dominant inheritance. And as I said, this mutation was already well known, and we were able to screen essentially this family for this mutation. I wrote in yellow below that no GC involved. It means that at that time, 20 years ago, when we did this study, it was part of my PhD thesis work, we did not have a genetic counselor involved to help us and to help the family understand what we are saying and how it can help them, and then take an informed decision whether they want to be tested or not. So we did the best we could. And we could not convince the family nor the clinicians to take any action based on genetic finding. If you look at the lowest generation, you will see there are three individuals, individual number 6, number 11, and number 13, where they are not filled in black, but there is a line going through the symbols. I am indicating by that is that these individuals were phenotypically normal, clinically normal, but they had the mutation, right? Just a bit of context in this family, anybody who had this mutation or did not have this mutation, they are absolutely normal until 12 to 13 years of age. It's only after that they started showing symptoms. And when they started showing symptoms, it was so aggressive that by the time they're 18 years old, they were completely blind, right? So you needed quickly act once they start showing any symptom. So we tried that. We tried to convince the clinicians and the family for these three individuals, but we did not succeed then. Next slide, please. Just to put it into context that what kind of families we are talking about. So this is the real house where these people live. They still live in these houses, right? I still go back to them whenever I can. I visit their village, and this is the actual house they live in. So you can understand their socioeconomic condition. It's a very remote, poor part of rural West Bengal. And imagine now a family lives in this condition where majority of the members are blind. Nearest hospital where they can get any treatment for their problem is about 2 hours away by public transport. So most often they don't come because they cannot come. A blind person alone has to take a bus, go to the hospital and come back. It's very difficult. And if somebody in the family who can see needs to accompany them, then they lose their daily labor pay, basically daily wage. So the most of the time the effect is that they don't go for checkup. So there is an accessibility of care system problem. Next slide, please. So what we did, so this have been agonizing me for a long time, that I know, or we can very easily, it's a very cheap and very fast assay. Those of you who have worked on DNA based assays know that isolating DNA from a blood or a saliva sample and doing an enzyme based digestion takes only about 3 to 4 hours or maybe a day at most. And it was very cheap. In Indian terms, it will cost you somewhere around 100 rupees per sample. And so what agonized me for a long time was that it was so easy for us to tell, and without any doubt, with full certainty, who will become blind later in life, but we were unable to do anything to help this family, right? So that kept bugging me. And then in 2023, only 2 years ago, with support from my current university, University of Salford, I decided to tell this story to the world in a different way. We decided to make a film. So what you see on this slide, on the left, you can probably see me and many others, where we went with scientists, and we went with filmmakers, social workers, and we sat down in front of their home in their village, and we developed a storyline where we tell their story true to its form without revealing their private information, without revealing their privacy, right? So we wanted to be authentic, but we also did not want to disturb violate their privacy. So we made a film, and on the right picture you see is that actually a scene from the shooting of the film, where we shot it in front of their house. The whole film is shot there. And so this film was made. Next slide, please. So this film is available on YouTube. The link is there. Many of you have seen the film. The film is called The Vision of the Blind Lady. And if you have not seen, I would urge you to go and see it. We tell the story of this family, the agony of this family, and also the hope for this family. In the next slide, I have a 2-minute trailer of the film, and I'll just be quiet for 2 minutes. And if you click on the bottom of the slide, a video should play. See? There's no audio. I don't know why the audio is not playing, but you can probably read the subtitles. That's okay. I think because there's no audio, we can just move on. Just click and move to the next slide. Sorry about that. I thought the audio will play. Yeah, so sorry about that. The trailer did not play as it should have. But anyway, I gave you the link to the film so you can go and watch it. So what happened then? Why I made a film and why I'm talking about it here and what the film did that other methods did not do. So for about more than a decade, I have been talking about this family, this case study, and I've been trying to advocate that, if for these kind of families, the rarer forms of glaucoma, of course, this is a rare occurrence. If you screen 100 patients, you will be lucky if you find 3 families like this, right? But for those 3 families, you can actually do something. The same principle with which ORDI functions, hence our collaboration in this particular project. And very surprisingly for me, once I told the story through a film, things started happening. So what has happened now? A change of practice started happening. So now we have a genetic counselor on board for this particular family. Dipanjana is, who you may know, is also part of this for the wider part of the activity, which I'll tell later on. So Emily has counseled members of this family, and those who agreed to be part of the study, they were clinically evaluated by Dr. Ashim Shil, again, one of the key stakeholders of this particular project. And once if they are found to be a suspect, or we suspected because one of the parents may have the condition, then the people on the top of the slide, Dr. Moinak Sengupta and Dr. Molinath Acharya, who are both based in Kolkata, they are geneticists, and they then came into the picture, and then they took the sample and did the genetic evaluation, confirmed that whether they have the mutation or not. If they have had the mutation, again, Dr. Emily Banerjee had done a post-test counseling, telling them what is the likely outcome if they have the mutation. And then those who agreed were undergone surgery as decided appropriate by Dr. Ashim Shil as the clinician, right? So this is what I've been talking about for more than a decade. And I've been talking about it as a scientist. I've been presenting papers, showing data. I was not talking about the human story. What the film has done is brought the human story to the fore, and that started changing things. Next slide, please. So not only these genetic counselors and clinicians came forward, also large charities like Rotary came forward. So specifically Rotary Club of Calcutta, Jadavpur, they are also a partner in this. What you are seeing here, the Rotary did a camp in the village where doctors went with equipments and screened them at their home. The genetic counseling sessions also happened at their home. And then Rotary again came in and solved the logistics of transportation, where they are brought into the hospital, those who are needed more care, more advanced care or a surgery. And then they're brought to the hospital, and then they were treated there. So all this started happening in the last 18 months. Next slide, please. This is just one example that how now, after 2 decades of agonizing over it, now we started to change lives as an impact of storytelling. So this is an example of a 13-year-old boy from this family. He is carrying this mutation in heterozygous form, as I explained, and he had a rapid increase in intraocular pressure in the eye during summer of 2023. And the difference that film has made is that without the film, they would be the clinicians would wait longer to operate on them, right? But now, thanks to the story that we told via the film, the clinicians now acted sooner, and now this boy had been surgically operated for both eyes before any of his vision was lost. So no vision was lost, only he started showing the initial symptoms of raised intraocular pressure. And this has been done almost 20 months ago, and he has been followed up for followed up every month for that. And today we are very happy to announce that he is free of the disease. The disease is not progressed, and what I understand from Dr. Ashim Shil is that we have a potential case of saved eyesight, and he is performing so well that he's not even having to put any eye drops even in his eye. So no medication is required any longer. And we will be continue to monitor him, but hopefully we have used genetics, precision health approach to diagnose and prevent a debilitating disease from happening in this particular child. And this part of the story, the success followed our first film, is now documented in the second film, which is called Ripples of Light. And that link is also given at the bottom of the slide. Next slide, please. So now this is the same family I showed you before. And I showed you, you can probably note that 3 individuals where I said that 20 years ago, I wanted to help them, but I couldn't, right? Just click once more. So these 3 individuals now, as you would predict, are now blind, right? Anybody who understands genetics would not be a surprise to them, but to the family, this came as a shock. The family did not understand. They thought, I am a magician. The family thought, who is not a doctor, when these children had no problem in their eye, how did he know that these people will go blind? So now they trust me more. Now click once more. Now these 3 people are parents themselves, and they have 3 children, right? The boy I showed you in the previous slide is one of these 3 boys here. And these 3 boys now are in the exact situation where their parents were 20 years ago, meaning they are around 10 to 12 years of age. They have absolutely no signs of the disease except the raised intraocular pressure that I showed you, but no vision loss. But all 3 of them are carrying the mutation. But now, with genetic counselor involved, as it says so on the slide, now we are able to change their life. Now in this generation, we have now, Dr. Ashim Shil has now done surgery for all these 3 children, and all of them are doing fine. Their disease seems to have been arrested, right? So what we are able to do, we are able to early intervene using surgery. But how that decision was made possible? The decision was made possible by use of precision health approaches, by use of genetics, where we found the mutation. So you could tell the clinician early on that watch this person closely. This person is going to have the disease. That's what the precision health is all about. So this we are able to do so far. Next slide, please. This, and if you want to know more, so I have a TED talk which is available in this link where I talk about it in more detail. You can catch me there talking about it from a different point of view. Next slide, please. But why does it matter? Why does it matter that we do this in genetic diseases, particularly in rare diseases, right? Why storytelling matters? Because, as it says on the slide, on average, it takes more than 6 years and nearly 17 doctor visits to receive a rare disease diagnosis after symptom begins, right? And if you see on the left of the slide, in a lifetime of a patient, the management cost is more than 200,000 US dollars. What the circle is showing, you see the left side of the circle, the dark blue color has more dots, and the right side on the light blue color has less dots. Less dots essentially translates to easier management and economically more viable. So, for example, for this family, if I take this example as I told you, that once the symptom starts showing, the mutation is so aggressive, so penetrant, that within 5 years they go from completely normal to completely blind. So they can't afford to wait the 6 years and 17 doctor visits with the remote location they're in. So if we can add genetic screening as to detect them early on, then we have the time. If we already know before they reach the disease of onset, if we already know who is going to have the disease, and we will also know who is not going to have the disease, because through genetic screening there's a 50-50 chance, as you may know. So as we'll pick out pre-symptomatic individuals, we'll also pick out individuals that are not carrying the mutation, and so we can direct the resources appropriately. So this is why storytelling matters. So because if I keep talking about logically, I don't make you feel, I can't make you feel what I have been feeling for 20 years. Go one slide forward, please. So when you hear stories, something very unique happens to your brain. Your feeling center connects to your logical center. Normally, when you're understanding science, understanding objective data, you're only engaging your frontal cortex, which is unique to us, unique to humans. But the frontal cortex cannot feel. It does not understand feeling. It only understands language and logical deduction and rationality. But when you feel, you engage a different center of your brain. The emotion, that part controls emotion. And that part does not understand language, does not understand logic, does not understand rationality, right? So storytelling is a unique gift that humans have. You might remember a story that your grandparents used to tell you 20, 30 years ago, but you may not remember a scientific article you read yesterday, right? Is because the grandma story made you feel something. So that made a unique connection in your brain. You can go back to that feeling anytime you want. A logical deduction does not give you that feeling. This is why storytelling is important. This is why the film achieved what me talking as a scientist did not achieve, right? And as one of my heroes, Dr. Brené Brown, says, the stories are data with a soul. So if you have the data and if you add soul to it, it becomes a story, and that moves people, that moves things. Next slide, please. So I'm towards the end of the talk. What I wanted to leave you with, thinking that this is a rare disease forum, rare info series, that the power of using the genomics or genetics for any rare disease where the outcome is more direct. That means the genotype to phenotype correlation is more direct. Those genomic studies are more actionable, right? So actionable genomics is different from predictive genomics. Yes, we can sequence whole genome today with a relatively lower cost, but we don't have to, right? Just because we have a hammer, everything is not a nail, right? We don't need to do sequence whole genome in every case. We can have targeted small scale assays because they'll be more affordable, more accurate, and more actionable. So we should have different strategy for different levels of genetic involvements. We do not need genome-wide screens for everything. My case study is a case in point. Next slide, please. So this is the summary. If you forget everything I said, I hope you remember this line, that if you add subjective storytelling, especially for humans, because human stories will move us quicker and better, with objective genetic data, if you add the soul to your data, then you can improve health equity in rare diseases. Next slide, please. These are the people. There are many, many people who have been with me, but there's a few key people I have to acknowledge. It all started with Professor Kunal Ray, who had been a PhD mentor for me, Dr. Molinath Acharya, and for Dr. Moinakshi Gupta. So it all started when I was a PhD student in his lab. I already spoke about Dr. Ashim Shil and Dr. Emily Banerjee, who have been instrumental in this journey as a clinician and a genetic counselor. And the other two people on the slide, Dr. Rumpa Gui and Shubhubroto De, are not scientists, not academics. They are filmmakers. So Dr. Rumpa Gui is a doctor in filmmaking, in storytelling. So they made the film. They are the directors of both the films that I talked about, and they have an organization called Seventh Art Communication, which made the films. The funding for the film and the funding for now continuing this work together with ORDI, together with Dipanjana, together with many other stakeholders in India, is funded by my current employer, University of Salford, Manchester. So it would not happen without them. I hold an affiliate position with CSR IGIB in Delhi, where I used to work before moving to Manchester. So they are still a partner in this journey with me. And also, many of you may know Premas Life Sciences. They are also a big believer in this journey, and they have been supporting me since the very beginning. Next slide, please. And I'm very, very excited. This is the last slide. I'm very, very excited that Premas and their sister branch Uncoded has now decided to come up with a genetic kit, next generation sequencing based kit for glaucoma. A disclaimer, I am no way affiliated with this company, nor I have any financial gain, but this panel, their decision to create this product in their portfolio has been catalyzed by our project. So that makes me very, very proud. And those who are interested, this is going to be launched next week during the Pediatric Glaucoma Conference in Madurai. And after that, that should be available to anyone in India. And it's a standard NGS assay, is adaptable to any of the Illumina platforms, and is much more affordable than a wider genomic analysis, and also simpler to analyze the data. So in this assay, where what they have done, they have put together probes to sequence 21 different genes, which are all known to cause glaucoma in a monogenic rare form. So these 21 genes will be screened together, and hopefully the case study I showed you will not be a singular event. Hopefully there'll be many, many more families across India who can be helped, and their blindness will be prevented in the future, that I can't wait to see. That is all from my side. Thank you very much. I'm happy to answer any question if there is.
Thank you, Arijita, for that wonderful, really talk. It actually summarizes, the entire work from bench to bedside. And in true sense, it can actually, we can see how, the change just does not need a lab thing, but it also needs to be translated into the grassroots level and the real grassroots level that we were talking about. Thank you so much for the presentation. And I don't think we have questions. As such, I can't see any questions in the panel or anything, but I'll from like so what do you think should be the way for screening in the general population, how do we get these patients, because these patients, many of them, as you said, will not come to the clinics. If they come to the clinics, maybe ophthalmologist or the retina specialist can take it up. But what is your strategy in India? What do you think should be the strategy in India to actually get these patients to the clinics itself?
Yeah, thank you for that. So that's of course the one of the bottlenecks in this whole work, and I think in a short term, what is already happening and hopefully will happen more wider once this kit is launched, if clinicians and genetic counselors, um, they actively advocate for these genetic screenings, if they actively advocate to the patients and how it can help. Obviously, everybody that will be screened will not be found positive for mutations in these genes. That's where genetic counselors are absolutely crucial in this whole process. So I think counselors should be able to explain to the family that what is the chance that they will find something. If they find something, what is the chance that it might happen, and it has to be catered from eye care centers, from ophthalmologists, right? So yes, the problem of transportation access is a bigger problem to solve, and that is probably my, I have a longer term dream in that sense, is that ideally we should be able to have portable kind of centers, that can mobile centers that can move to relatively remote areas, and maybe after consultation, if they agree, they take their samples, carry the samples back to the lab, so that we can at least do the screening even without the eye test, so that then we can report back to maybe the local health centers that these are the people who should be monitored closely, because they are going to they are at a much higher risk of having glaucoma. So it is a long term public health effort almost, and this is a big challenge, and we need many stakeholders like yourself, like many other ophthalmology, who has to come together and make it happen. So that's my answer.
So, when you say about these tests, can you give us an estimate of what would be the cost of these screening tests to the like the
I
patients?
I can't. The reason I can't is because as I said, I'm no way involved in marketing or commercializing this product. If I say something, it might be very wrong. I will only say that when the company kind of told me, they told me because the work inspired them to make this product. And my only request to them was, please keep the price much lower than the whole exome tests that are currently available, and is kind of, at least in big urban centers, it is pretty much commonly getting advised, not only for glaucoma, for many other conditions. So it should be much lower than that, and they have said that they will keep that promise. That's the only thing I can say, but I don't want to put a number because I can be grossly wrong. So yeah.
No, because you said it's a public health thing. So I was thinking, like, how do you feel the government will chip in? Like, will it be covered by insurance? Because most of the genetic testing in India are not. And if we have to, if even if somebody is, giving the samples, there is consent. There is obviously data protection. There is obviously a lot of other things to be worried about. So, like, your thoughts on that?
My my this can be a naive response. Okay, this can be a naive response, having now worked with ORDI in the past few months, talking many sessions with Prasannaji. I now see my naivety, but my naive thought, which I still have, I'm trying to find a way to have diseases such as these, which does not by definition does not meet the rare disease criteria, but to bring them under the rare disease umbrella, because the difference that I see is, for cases like these, the rarer form of common disease, the cost for their management is far less than cost of an otherwise classically known rare disease, right? The lifelong costs are far less. So if that can be done from a financial point of a public health burden point of view, that's probably, in my mind, is the lowest hanging fruit, right? Knowing whatever I know, my knowledge is limited, how things work in India at the moment, but knowing what I know, thinking that it will be in the near future come under coverage by insurance, is probably a much more complicated ball game, right? So I think that if we can redefine what we mean by rare disease and bring these type of conditions under that umbrella, and maybe allocate a pot of money, not as much as other rare diseases need, that probably is my way to go for it. But again, as I said, I know partly that it's a naive thinking.
Thank you so much, Professor Arijit Mukhopadhyay, to give us this beautiful, wonderful insight into into how you think this will work, and it's actually a work from, as I repeat myself, from bench to bedside, and absolutely at the grassroots level. It was a pleasure to hear you. I thank all the audience and organization of Rare Disease India and Prasanna sir for giving this platform for us to express this. And we will be back next week with another such session to bring in insights. And with that, I leave all of you with a very, very good evening. Stay warm, because it's the temperatures are kind of nipping here in everywhere in India. So until next time, thank you so much.
Thank you. Bye bye.